Welcome back again online. Thank you so much. You've been applauding online for Mr. van Vollenhoven. Lovely. Even someone sent in new 100-year-old music for him to give to him. So that was very lovely. Thank you for participating and thank you for voting all the time. You can use the chat, the Q&A, whatever, because we really like knowing that you are there. Thank you also all the young females, especially, who came to support me on my cleavage. Wonderful. Love that. I agree with them. It's lovely, right? Why not? What do you think about cleavage, Ronald? All right. I have with me the director and the team leader of CNCR, the Center for Neurogenomics and Cognitive Research. And I heard, Guus, Ronald told me that you, in the COVID time, that you too tried to stay at home as much as you could, so other people could be in the lab.
Yes.
More.
Yeah, we had to do some hard decision making, obviously.
Yes.
But to not occupy the lab space, basically, those that could stay at home stayed at home and worked from home, and those that really needed to be in the lab, of course, went into the lab. Yeah.
Yes.
So that's how we tried to solve and cope with it. Yeah.
Yes. So you were just a few days there, and for the rest of the time you were at home.
One day even.
Yeah.
One day in the lab.
One day.
Home,
yeah. That's not a lot.
No.
So what was the resilience challenge? What was the most important challenge in resilience for you?
Well, I think the most important in such a time is to keep all those people in the lab together, basically. You know, so we really made an effort also to have our lab meetings and our seminars ongoing, and etc. Right. So keep all those that are working from different locations together. Right?
Yes.
And that helped a lot, I think.
Find togetherness.
So we kept communicating.
Yes.
Because we are social animals, right? So we need to work together.
Yeah, we are. Okay, thank you so much. So you're going to present now first. That's lovely. And later on, there'll be a keynote speaker, Li-Huei Tsai, and you will introduce her and we will talk to her. But for now, I'd say, please take the stage. Yeah. And of course, Ronald van Kesteren. And of course, you can enter your questions in Slido, and you can raise your hand here if you want to ask a question live after, or make your comments. Everything goes.
Yeah. So actually, all I want to do is also to sort of introduce our main speaker of today, Li-Huei Tsai. I will show you some of my own work, but it's not that important. It's all just an introduction to Li-Huei Tsai, I would say. So the main point here is how can we recognize Alzheimer's disease at an early stage? So Alzheimer's disease in particular has a very long preclinical stage. And that preclinical stage, it's important to recognize that as early as possible and also be able to treat it as early as possible. And we have a lot of markers that we can use to recognize the cognitive state of the disease, really diseased people. But when it comes to this early stage, there is one that stands out at the moment, and that is early neuronal network dysfunction. That has become very popular over the past decade. People have looked at that in patients. People have also looked at that in animal models. And that is hopefully also where the two at some moment might meet and instruct each other how to get better markers that can predict Alzheimer's disease. So my focus is on networks. And when you look at networks from a fundamental perspective, so how can you study neuronal networks, neuronal circuitry in an animal model, then you really also can look at the individual cells that contribute to network activity. So network activity is generated by the principal cells in the network, the excitatory cells in the network, which in many brain areas are pyramidal cells, the ones that you see over here. Those are the excitatory cells. They generate activity. But what is important to realize is that there's many different types of inhibitory cells, interneurons, that actually shape network activity, that determine how network activity looks, that pattern network activity. And we as a lab are interested in what these interneurons have to do with network activity and how they are involved in Alzheimer's disease. And we focus on parvalbumin interneurons in first instance, PV interneurons. But I will also briefly mention somatostatin interneurons, and that they all have been shown also in other papers to be very important in Alzheimer's disease. So my lab is interested in that, and one former PhD student in my lab, Li-Huei Tsai, started to wonder what is going wrong with, in particular, these parvalbumin interneurons in the early stages of disease. So we use animal models for that. We use an APPPS1 animal models, which has 2 human mutations introduced in the animal in order to generate increased amyloid production, which is a key feature of Alzheimer's disease. And one of the things that she then started to do is to look at the electrical activity of these parvalbumin neurons very early in this animal model. And what she found is that these cells are actually become very early, they become hyperexcitable, they become hyperactive. So you see that over here in that they have an increased resting membrane potential, and in these curves you see that they have an increased firing of action potential, so they are hyperactive. And then Sarah realized that, and that is one of the great things of doing fundamental research, we have tricks, we have genetic tricks in order to be able to specifically also manipulate, restore activity in these cells. So Sarah used, sorry, I'll skip that one. Sarah used a chemogenetics for that. She introduced receptors specifically in parvalbumin cells in the hippocampus, and then she was able, and this is just the summary is the only thing that's important. Follow the orange arrows. She was able to restore the resting membrane potential in these cells. She was also able to restore the firing pattern in these cells, and she was also able to restore inhibitory transmission in these mice. And so the network, at the network level, we were able to restore everything back to normal in this Alzheimer mice by simply rescuing the activity of parvalbumin cells. And importantly, memory is of course a big issue in Alzheimer's disease. That is how you should read out whether this is really also a functional restoration. And so we measure memory in a Morris-Water maze, and in a Morris-Water maze, mice need to find a platform, a hidden platform, and they need to locate it, and you can measure whether they have memory for that location. If you then follow how they swim in that maze, you see mice that have memory swim always close to the platform location. Mice that don't have memory forget where the platform was, and they just swim around. And also this was completely rescued, as you see again with the orange arrow, was completely rescued if we restored parvalbumin cells. So today is about resilience, and I want to show you one other set of data that Sarah produced in my lab, and that is this trick that we used, this chemogenetic trick to reduce the activity of parvalbumin cells. You can use exactly that same trick also to increase the activity of these cells. And that is a bit of a silly question because that would generate Alzheimer's disease, but in an animal model that's interesting to test. So we can do that in wild-type animals. So we reverse the trick. We now increase the activity of parvalbumin cells, and we do it in a healthy condition, in a healthy mouse, in a wild-type mouse. And then you create a network condition without the presence of amyloid, a vulnerable network condition without the presence of amyloid. So indeed, what you see then, we do that in a healthy situation where the circuit is in balance. There's excitation, inhibition, balance. And the memory, these cartoons here underneath, they indicate the memory performance, and these animals have normal memory. So if you increase parvalbumin cell activity in these mice, you see that the inhibitory component in the excitation-inhibition balance increases, and this initially impairs memory. However, we see that soon thereafter, the circuit tries to restore itself, actually, by then increasing also the activity of the excitatory cells. So you restore excitation-inhibition balance, but at a higher excitation and a higher inhibition level. And this seemed to be very good for the memory. These mice have actually enhanced memory performance. Except when we trigger this system, when we challenge this system with a low concentration of amyloid, a subthreshold concentration of amyloid, then this hyper circuit all of a sudden is very vulnerable, extremely vulnerable to such a stressor, beta amyloid in the system, and the system collapses in a very Alzheimer-like way. So the parvalbumin cells get impaired. You see overexcitation in the circuit, hyperexcitability in the circuit, and impaired memory again. And this is actually my only connection today to resilience. I mean, this would indicate that network operates at an optimal level of resilience. So you can have EI balance at different levels, at different offsets. But the actual EI offset that you have may determine how Resilient neural networks are against stressors such as amyloid. If you have suboptimal EI balance, the system may actually not be sensitive enough to work at all. But if you have superoptimal EI balance, that may be very beneficial for cognitive performance, but the system may be very unstable, very vulnerable to stressors like amyloid. So how relevant is this all now for the clinic? PV cell dysfunction we cannot measure directly in Alzheimer's patients. That's a problem, of course. But we know that PV cells are involved in generating particular network oscillations. And PV cells in particular are important to generate gamma oscillations in the network. In mice, you can measure that in the LFP signal, the local field potential signal. You can measure gamma oscillations. In humans, we measure that with EEG or with MEG. And it's, of course, interesting to now try and find out whether those two are connected, whether we can see anything in mouse LFP data that resembles what we see in early stages of Alzheimer's disease, and whether we can use mouse data, in particular also the underlying cell physiology, to better interpret, to better understand which markers in there are relevant to the prediction of Alzheimer's disease. So that is something that we try to do currently. I want to show you very quickly just a few data from another PhD student in my lab, Ronald van Kesteren, who is setting up doing LFP recordings longitudinally in living mice in order to measure in different brain areas, prefrontal cortex, parietal cortex, and hippocampus with different electrodes, LFP signals that can generate oscillations in the same way as you can measure them in human EEG data. So currently what we have, it's very preliminary yet, and Fran is very busy in analyzing all this data. Currently what we have sort of follows, aligns nicely with the fact that we see hyperexcitable parvalbumin interneurons, because we indeed see in some of these brain areas increased gamma oscillations. Interestingly, we also see increased beta oscillations in the LFP signals, which suggests that maybe some of these other interneuron subtypes, and I mentioned already somatostatin interneurons, may also be involved because these are the generators of beta oscillations. So we are looking into that, whether how other interneurons are involved. And it all seems to go at the cost of the lower frequency waves. So we also see a significant reduction of delta oscillations, for instance. So there's an overall acceleration of the profile. And so this may help. So knowing the cell physiology underneath this in mice may help to find what is a more predictive EEG biomarker or MEG biomarker later in humans. Finally, we also are interested in treatment targets. And Fran is also doing single-cell RNA sequencing on these neurons in order to find out what are the molecular changes that we see in these neurons that may underlie these changes in network activity, and can these be used as treatment targets. And one of the interesting things with single-cell RNA-seq is that you get per cell type, you get an expression profile. And one of the things that we noticed is that in particular in interneurons, and only in interneurons, there seems to be a dysregulation of several potassium channels, which are very important in generating electrical activity. And these are interesting to follow up, I guess, because it's known already also from other clinical studies, other disease areas actually, that if you modulate potassium channels, and there are specific inhibitors and activators of these channels, that you can actually modify the type of electrical activity that we see disturbed also in these neurons. So in summary, I think the most important thing that I wanted to tell you, and again, it's all in preparation of what we are going to listen at, I guess, in the next talk, where we also will see clinical applications of manipulating network activity. I think that what I hope to show you is that knowing the cell physiology underlying brain oscillations can be very important in a translational approach to come to better clinical markers for Alzheimer's disease. And also looking at the molecular profiles in these cells may come actually at targets that you can treat in order also to treat the network at an early stage. And these are the people that contributed. Most important, Sarah Hijazi, Frans van Heusden, who did all the work. But here's my other collaborators that are involved in here, and also my funding that I received for this particular type of research. Thank you.
Yeah. If you have a question or a comment, this is the time. Then please raise your hand, of course. There you are. Let me see, I have the microphone here. Okay. Yeah, you have to stand again for the online participants, if you can. So
This is very exciting. So we see the connection now with Li-Huei Tsai, but she is going to show us a story where actually gamma band oscillation is actually presented as a cure. So how do you relate to that? Just by looking at it in advance. And so does this have to do with hub vulnerability in networks?
Yes, very also. So then you should have to know how gamma band oscillations are related to cognitive processes, and also how they are synchronized with other types of oscillations, I think. But gamma may be a hub in cognitive processing, I agree. So it's a very elegant way of like try to manipulate the network and see whether you can, at least the cognitive components can also be manipulated in that way. I think a better understanding of also the other changes and the other neurons involved may also in the end lead maybe to pharmaceutical targets that you can use instead of the simple gamma frequency stimulation. Yeah.
Thank you.
Hello, my name is Wilma van den Burg. And I have a question in the context of resilience of the topic of today. You mentioned that these parvalbumin neurons are very important in terms of how they are active. Is it a local network in these regions that determine their resilience, or is it more on global level how they are connected?
Yeah, it's locally how these cells generate gamma oscillations, but of course the synchrony of gamma between brain areas is also important. So we don't know yet, but we are looking into that. So this is all in the hippocampus, but are now PV neurons also in, for instance, the frontal cortex? Are they also affected? And if not, is it like the communication, the gamma synchrony between these areas that is then affected? So can you have like affected synchrony even if in some areas parvalbumin neurons are still intact? That's type of questions that we still try to address.
That's exactly why I've asked it, because I can imagine that the local circuitry in the local network in these regions, they're different on cellular basis, and that could have a direct effect on how this resilient or how they react to our beta.
Yeah, exactly. So we are currently looking also in the prefrontal cortex in order to understand that are PV cells there also affected or not, and if not, is there another mechanism, communication mechanisms, via gamma or otherwise, that determine then why networks collapse.
Thank you so much. Thank you so much. You already said this was kind of an introduction towards Li-Huei Tsai's story.
Yeah.
Okay, and maybe you can add a little to that. Yes, you take a seat and you'll join us, and we'll talk to her. We will talk to her later live, and then,
we will.
Then she'll be in Zoom, and then you can ask your questions and talk to her. But for now, it's a recorded presentation, right?
So Kim, we are looking forward to that, and you know, I think you all sometimes have a paper that comes out in the field, you know, and then you think this is really something, you know, and that is what we had with Li-Huei Tsai's paper a few years ago when she published in Science on a topic that is really related, of course, that Ronald shows, and that showed, in fact, that these gamma oscillations that you can interfere with, as Ronald showed, at the cellular level, but then you have to manipulate the system, for instance, in animal models, that you can also do that from the outside. So she showed, in fact, that the gamma oscillations could be changed, and you could rescue, in fact, gamma oscillations also from the outside by using a 40 Hertz light trigger, you know? So that's a non-invasive way of intervention in the brain, you know? And we thought this is so cool, we should show you. So that's why we invited her, and just give me 10 seconds or so to mention a few things about her. So she did of course, her PhD and her PD postdoc that she did in Cold Spring Harbor, but at the same time doing that fundamental research, she was already in the Massachusetts General Hospital, so she has always this connection between fundamental research and clinical research. That's really interesting. She moved then to Harvard, Department of Pathology, which in a way did not completely satisfy her research needs. So she went back, in fact, next door to the Picower Center, where she is also today. And there she also does her research. So that's from 2006 onwards. She has many, many awards. I'm not going to talk with you about that. She has many outstanding papers. For those of you who are interested, look into it, because there are many different topics that are related to Alzheimer's disease, not only the one that she shows today. They are in Cell Science, Nature Neuroscience, Neuron, all in the last 3 years. So you can enjoy. Kim, I think we're ready to look at her presentation.
I think so too.
Okay, good.
Hello, everyone. I'm Li-Huei Tsai. It is a tremendous pleasure to be here today to tell you about our most recent progress on using non-invasive sensory stimulation to entrain the brain and to provide neuroprotection. So now I'm going to share my screen with you. So this is my disclosure slide. I would like to particularly point out that our technology has been licensed by Keynote Therapeutics to go forward with human clinical trials. So we all know that there are many different classes of brain oscillations or brain rhythms in the mammalian brain. And these different classes of rhythms cover several order of magnitude of frequencies from the infraslow to the ultra fast. And these different classes of brain rhythms are highly evolutionarily conserved from animals with very small volume of the brain, such as the mouse, to the primates, such as humans. And today I'm going to particularly focus on the so-called gamma rhythms ranging between 30 to 80 hertz. So gamma oscillations are known to associate with higher order cognitive functions and is important for rapid information processing. So increased gamma oscillations is known to occur specifically on a particular behavioral state, such as during working memory, sensory processing, and spatial navigation. But there are also many things we don't know about gamma oscillations, especially at the molecular level. So when there's increased gamma power, what happened to neurons or other type of cells? And also, how does gamma influence other cell types if there are effects there? So today, I hope to be able to answer some of these questions. An important facet about gamma and that got us interested in looking into gamma is the observations that gamma power is compromised in Alzheimer's disease, both mouse models and in human subjects. We reported previously that even in the pre-symptomatic stage of amyloid mouse models, such as the SXFAD mice 3 months old, in the hippocampus, when we perform local field potential recordings, we found that gamma power is already reduced, and this gamma power recorded centers around 40 hertz. And also, this is a list of mouse models that have been shown to have reduced gamma power. So just a few words about Alzheimer's disease. We know that Alzheimer's is the single disease that really can represent the biggest threats against our increasingly aging society. There are more than 45 million people worldwide afflicted by Alzheimer's disease. The disease is featured by the pathological features, including amyloid plaques, neurofibrillary tau tangles, as well as cerebral volume loss, synaptic loss, and cognitive impairment, eventually leading to dementia. Decades of research revealed various disruption at the subcellular and subcellular levels. Many brain cell types are involved, and many human genomic loci have been implicated as well. However, despite all of this research, we still don't have effective therapeutic intervention that can either prevent or cure the disease. So as I told you earlier, the gamma power is compromised in Alzheimer's disease. So initially, we wanted to just test the idea to increase gamma power in this Alzheimer's disease mouse model and see what happens. So my former graduate student, Hannah Alcarino, in collaboration with Ed Boyden and Annabelle Singer, we use optogenetics. So we specifically drive the albumin positive interneurons in the hippocampus, expressing channelrhodopsin 2. So we use 40 hertz laser pulses. And what we found is a tremendous increase in 40 hertz gamma power. But the most surprising results at that time, I remember when Hannah showed me, is that after 1 hour of stimulation, she found that in the hippocampal lysates, there was tremendous reduction of beta amyloid peptides, both A-beta 40 and A-beta 42. And this reduction was very specific to 40 hertz, not seen with 8 hertz stimulation or random frequency stimulation. So when we presented this work at our institute, Emery Brown, a colleague and a physician, pointed out that optogenetics is quite invasive, and to have any clinical implication, we should think about any non-invasive approach to increase gamma power. So we looked into the literature and stumbled upon this beautiful publication by Wolf Singer. This was more than 20 years ago. They exposed cats to light flickers. So they use a range of stimulus frequency anywhere from 0 to 50 hertz, and then recorded the response frequency in the visual cortex. And what they found is an absolute linear relationship of the stimulus frequency and the response frequency, indicating that light flicker can induce rhythmic neuronal firing. So, so we wanted to know whether this could work in mice, knowing that mice are much less a visual animal. So this is Hannah Alcarino. She, at the time, decided to expose mice to LED light strip, and the strip can flicker controlled by an Arduino system. So, when she exposed the mice to 40 hertz light flicker, we could record a sharp increase in the LFP power at 40 hertz in the visual cortex. So we turned this approach gamma entrainment using sensory stimuli or GENUS, because really sensory stimulation could entrain neurons in the brain. So then Hannah did the following experiments. She exposed the mice to the light flicker for 1 hour at different frequencies, and then evaluated amyloid peptide concentrations in the visual cortex. And what she found is that, very similar to optogenetic stimulation, in the visual cortex of the 3-month-old SXFAD mice following 1 hour of 40 hertz flicker, there was, again, huge reduction of A-beta 40 and 42 peptides. None of the other control frequencies led to reduction of A-beta. So these results clearly show that sensory stimulation induced brain entrainment can have biochemical consequences. So how local or widespread can GENUS induce gamma entrainment in the brain? China Daikan, a postdoctoral fellow, decided to record LFP power in multiple regions of the brain using a custom-made macro drive. And so he turned on 40 hertz LED light flicker and then recorded in the visual cortex, somatosensory cortex, hippocampal CA1, and the prefrontal cortex. And I think you can see the results. Visual stimulation can lead to a widespread entrainment throughout multiple regions of the brain. Moreover, China decided to see whether the biochemical changes can also be induced by sensory stimulation. So in this experiment, he exposed much older 5-SFAD mice, 9-month-old mice, to chronic GENUS. This involves 1 hour per day of stimulation for a total of 3 weeks. So in the 5 months, in the 9 months of SXFAD mice, you can see amyloid plaques, widespread amyloid plaques deposition throughout the brain. But after 3 weeks, 1 hour per day of GENUS, amyloid plaque is reduced in all of the regions examined. And this reduction of pathology is not restricted to plaque. He also applied chronic GENUS to a tauopathy model known as the P19, P301S transgenic mice. And here, using a phospho-tau epitope-specific antibody, you can see in the control mice, there is this very obvious tau aggregation in many neurons. But after 3 weeks of GENUS, this tau pathology is greatly reduced, again, in all of the brain regions evaluated. Finally, I told you earlier that there is tremendous neurodegeneration and brain volume loss in Alzheimer's. We have a transgenic mouse model known as the inducible CKP25 mice, where the P25 transgene is very neurotoxic. And upon induction for several weeks, such as 6 weeks, there is widespread neurodegeneration, lateral ventricle enlargement, and neuron-positive neuronal density loss. So again, we treated these mice with GENUS simultaneously with P25 induction for 6 weeks. And we found that GENUS was able to prevent neurodegeneration induced by P25. This is really quite remarkable. We've never seen anything like this. So the lateral ventricle enlargement was very much reduced, and neuronal density is restored in the visual cortex and in hippocampus CA1. So far I show you that we were able to use visual stimulation to entrain the brain. Subsequently, my former graduate student, Anthony Martorell, together with Ed Boyden and Annabelle Singer, we came up with a algorithm to stimulate mice with auditory gamma stimulation. And we found that this stimulation can similarly entrain neurons in the auditory cortex and hippocampus. And we further combine the auditory and visual stimulation together to show that this combined modality stimulation can facilitate propagation of the whole brain entrainment. And I really meant whole brain, because here you can see the clarity shield process brain in the 5-SXFAD mice. The amyloid plaques is widespread, not just in the cortex, but also in the deeper parts of the brain. But following the combined modality stimulation, you see a brain-wide reduction of amyloid plaques, not just in the cortex, but also in the subcortical regions. So knowing the profound effects of GENUS on the biochemical changes and reduction of Alzheimer's-related pathology, we wanted to know whether this can have functional consequences as well. And to cut a long story short, we were able to show that chronic GENUS stimulation can rescue a plethora of different behavioral deficits. This is an example to show you using Moore's water maze task to evaluate spatial learning and memory capacity in all of these different transgenic animal models we tested so far. We saw improved spatial learning and spatial memory performance in all of these models. So you may want to ask me, so far everything sounds too good to be true. What are the mechanisms underlying the beneficial effects of GENUS? So this is the one question that many people in my lab are addressing currently. We don't have the complete answer, but I can tell you what we know so far. One is that the brain's innate immune cells, microglia, respond robustly to GENUS. So this observation was made several years ago. We found that following visual flicker stimulation, microglia in the 5-SFAD mice completely changed their morphology compared to the control animals. So this green labels are aberrant staining of microglia. So you can see this microglia. So body volume is tremendously increased, and also the cell look much more ramified. But it's not just the morphological changes that we can observe. We also perform RNA sequencing to report there is significant changes at the transcriptomics level. So we isolated microglia and neurons from treated mice and perform bulk RNA-seq. So in microglia, what we observe is a tremendous reduction of genes involved in inflammation and immune response, and upregulation of genes involved in vesicular transport and catabolic process. Also in neurons, we observe downregulation of apoptotic signaling pathway, DNA damage response pathway, and upregulation of vesicle-mediated transport. You can see a repeated cellular processes that are influenced by GENUS and synaptic signaling. So in addition to neurons and microglia, we found that the vasculature in the brain also responded. So here, Mitch Murdoch is using 2-photon live imaging, real-time imaging to look at the blood vessels. He used a fluorescent dye to label all the blood vessels in the mouse. And then he trained the mouse to run on a wheel. And then he opened a cranial window in the skull of the mouse, so he was able to visualize the capillaries. So what he found out when he turned on the 40 Hertz LED light, the capillary diameter is significantly increased in every single animals he evaluated. Also, the velocity of the blood flow is also tremendously increased. Mitch now is particularly interested in another brain waste clearance mechanism known as the glymphatics. So the glymphatics really is known to describe CSF influx into the brain from the subarachnoid space into the brain, traveling along the descending arteries. And eventually the CSF reaches the capillary beds and exchanges with the interstitial fluid, and in the process facilitates brain waste clearance. So Mitch was able to show, using a CSF tracer introduced into the cisterna magna, that the CSF, according to the time-lapse imaging, indeed travels into the brain along the blood vessels. But most importantly, with 40 Hertz sensory stimulation, there is a tremendous increase of CSF influx compared to the animals without 40 Hertz stimulation. And this increase, again, is frequency dependent. It's not that just any neuronal activity stimulation leads to increase in CSF influx. Only 40 Hertz stimulation increase CSF influx, but not 8 Hertz stimulation. And he figured out that vasal motion is most likely to mediate the 40 Hertz induced CSF influx. So vasal motion refers to the dynamic motility of the arteries, and this is speculated to mediate CSF influx. And he was able to show that after 40 Hertz stimulation, the vasal motion is tremendously increased, and this is not affected by 8 Hertz stimulation. So just to summarize what I told you so far, I show you that 40 Hertz sensory stimulation can elicit response from multiple cell types in the brain, including neurons, microglia, astrocytes that I didn't have time to tell you, as well as the vasculature and the lymphatics. And we believe that it's the combination of the response from all of these different cell types resulting in reduced Alzheimer's-related pathology and improved cognitive function and synaptic function. So in the last several minutes, I would like to tell you about our effort in testing the potential effects of GENUS in human subjects. So this effort has been led by Dr. Diane Chen, a MD PhD neurologist. So knowing how non-invasive GENUS is, we went ahead to come up with a device where we can deliver to our human subjects. So this involves a 2-foot by 2-foot LED light box and a high definition soundbar. So this device can deliver both visual and auditory sensory stimulation. We also initially performed EEG recordings to evaluate entrainment in human subjects. So I wanted to show you that we initially tested our device in younger cohorts, older cohorts, and an Alzheimer's disease cohort. And here you are looking at the topographic map of the skull, indicating increase in 40 hertz RFP power. And you can see, compared to visual stimulation, auditory stimulation, the combined visual and auditory resulted in the most profound increase in 40 hertz gamma entrainment throughout the brain. So this is the back of the brain, the visual cortex or occipital lobe, and the top is the frontal lobe. So, so this profound increase in 40 hertz power occurred in the young group, but also in the older group and in the Alzheimer's group. I should also say that this approach appears to be extremely safe. There is not a single incidence of seizure that we observed. Also, there is not even complaints of headache or changes of vision or hearing function. And the subjects usually claim that they feel calmer and they feel very energetic following the stimulation. So, so with that data, we initiated a Alzheimer's disease small-scale trial, efficacy trial. We recruited 15 early-stage Alzheimer's disease subjects with average AMSE score of 22 to 23.5. So 7 of them were in the control group receiving constant light and white noise, and then another 8 were in the active group receiving 40 hertz light and sound stimulation. I should say that we install our device in their home, so they are able to turn on the device to treat themselves on a daily basis, and they're supposed to be exposed to the light and sound stimulation for 1 hour per day. And also, we initially designed the trial for 1 year long with evaluation every 3 months. But unfortunately, the pandemic happened. I'm going to be able to show you the data at 3 months time point, because after that, MIT was closed and we were no longer able to bring the subjects in. So after 3 months of exposure, when we use volumetric MRI to look at their brain volume, the control group continued to show expansion of the ventricle, but the active group seemed to show much less expansion. And also the hippocampal volume loss, this is in the control group, but in the active group, they seem to be more stable. Also, we perform functional MRI measurement to look at the posterior cingulate connectivity, as well as the medial visual network. And here you can appreciate these differences are actually pretty dramatic. In the control group, we saw there's continuous decline of the PCC network and the medial visual network, but in the active group, we actually see an improvement. So finally, we perform one functional task, which is known as the novel repeated face name association task. We projected different faces with names to the subject when they're in the MRI chamber. And then after they were out of the chamber, we showed them the name and asked them to associate the name with the name. We showed them the face and then asked them to associate the face with the name. And we found that the active group subjects show significant better performance than the control group. And very strikingly, the performance on this task has a very, very positive correlation with the medial visual network functional connectivity. And finally, I would also like to mention that another group at Emory University and Georgia Tech, led by Annabelle Singer, who is a PI there now, also performed a human small-scale feasibility trial. And they also perform functional MRI study and look at the PCC network. And they evaluated only after just 2 months of daily exposure. And there they also saw a significant improvement in the PCC functional MRI network. So in summary, I show you that GENUS and transhuman subjects safely, and the combined A+V modality induces the most effective entrainment, and GENUS prevents enlargement of the lateral ventricles and hippocampal atrophy, and enhances functional connectivity in the PCC and medial visual network, and GENUS improves memory function as evaluated by the face name association task. With that, I would like to thank all the people contributing to the work. I mentioned Hannah Alcarino, Anthony Martorell, China Adaiken, and Mitch Murdoch. So Diane Chen is leading the human gamma team, and Ho Jing Suk and Brennan Jackson contributed significantly. I would also like to thank my collaborators Ed Boyden, Annabelle Singer, Colin Chung, Emery Brown, Brett Dickerson, Keith Johnson, and Aaron Bose. Finally, I would like to thank my funding agencies for their support over the years to make this work possible. Thank you for your attention.
Yes. So, we have many questions coming in, and I see a hand here already, so there are many there. First of all, if you take a look at Slido, you can vote for the questions that you like, so that we are sure that the most important ones will be on the top, so that with the little time that we have, we will be able to ask Li-Huei Tsai all the questions that really matter. Now, I think she's here now, but I have to know, I never checked, what does she see? Where do we have to look at her, Gerdy? In that camera? What does she see? Do you see us? Yeah. Hi. What? Yeah. Yes. That was the round of applause for your presentation. Yes. You have been introduced and we've been listening to you, and questions are coming in, so welcome, welcome. Nice to have you with us. I'd say that I go into the room, collect questions, and that you read out the top ones of the Slido questions. Yep. So they will probably show at the monitor. Is that possible at the same time? Yeah, that's good.
Okay.
Go ahead.
Let's see. First question, Wiesje van der Vlier. Wiesje asks, I would expect that oscillatory brain dynamics are the consequence of structural brain changes, for instance, amyloid deposition. Your findings suggest the opposite order. What would be the mechanism? Interesting.
Yeah, I mean, this is like a chicken and egg question. I agree that amyloid deposition probably will change the dynamic of brain oscillations. But our experimental results clearly show that if you can increase the power of gamma oscillations, you can mobilize all these different mechanisms, physiological mechanisms that normally functioned to clear brain waste and maintain the health of the brain. So, I think that that's why our approach of using a very non-invasive approach to boost the oscillation in the brain is very promising.
Okay. Thanks a lot. Another question for Sabine again, yeah?
From the audience.
Yes.
Hello, Sabine Spijker. Nice to meet you there, and thank you for your presentation. So my question was related to the longevity of the effect. So once you do this, say, 3 weeks of GENUS approach, how long do you see the good effects? And is it also safe to use for controls? So like control mice, control humans?
That's a great question. So I should say that right now in the animal model, the longest treatment we have done is 2-3 months. And then if we stop the stimulation, I think we can continue to see the beneficial effect lasting for a few weeks, but eventually it wanes off. So that suggests to us that it is necessary to do the stimulation regularly. In terms of stimulation of the wild-type mice or in healthy humans, so far, we really haven't seen much of an effect. In young, healthy mice, I think normally their endogenous gamma oscillations is already very, very strong. So the additional boost, at least in our hand, did not seem to impose a measurable phenotypes. And in humans, we have also recruited healthy subjects, and they seem to tolerate the stimulation well.
Thanks. Guus, another Slido question maybe?
Yeah, maybe we can put up the questions. Yeah. Do you think the effects you're seeing are acute to the treatment or can be long-lasting? Well, that's in fact close to Sabine's question, I think. But yeah, I think we have had that question.
I can tell you, we were very surprised from our recent experiments. So initially, we always thought that we needed to do very chronic stimulation for weeks or something to see meaningful reduction of pathology. But just recently, we realized that even with acute section, the reduction of pathology is already measurable, at least in animal models. So that's something we're still investigating.
Okay, yeah. Thanks. We have a question here. Maybe we can say hi, your name to.
Hi, my name is Priska Leverink, and thank you very much for your presentation. I actually have 2 questions, if that's allowed.
The most important one.
So how specific do you think the GENUS stimulation is for specifically clearing amyloid beta? And do you think it could also apply to other proteins that accumulate in the brain and are associated with neurodegenerative diseases?
That's a very interesting question. So we definitely show that it cleared phospho-tau pathology and tau aggregation. We so far have only worked with Alzheimer's model and looking at Alzheimer's specific protein aggregation. But I think it would be interesting to see whether pathology associated with other neurodegenerative diseases can be affected as well. In general, I think that since GENUS recruits very fundamental mechanisms such as microglia activation and vasculature dilation, increased blood flow, and now we have some evidence indicating CSF influx. I would speculate that the effect could be more general.
Thanks again. And we have time for a Slido question again. There are so many. Sabine wanted to ask another question. But maybe we can transform this into a storm of question and answers. So short question and answers, because people are so interested, Ms. White. That's great.
Let's do a short one. Does it matter whether visual and auditory 40 Hz stimulations are in phase or out phase?
Yeah, we so far have been doing synchronized visual and auditory stimulation.
Okay, that's a short answer. Now jump up. You can ask your question.
Okay, so would it also work for other type of diseases where EI imbalance is affected? So like schizophrenia, depression, these type of diseases.
Thanks.
Yeah, that's a very interesting question. So far, from our behavior studies, we find that definitely animals have reduced anxiety level. So beyond that, we haven't really looked at schizophrenia specific behavioral task. So I cannot say that, but I think it would be worthwhile testing, especially since the approach, again, is very non-invasive.
Ronald, maybe you can pick one of the Slido questions.
One Slido question?
Oh, you want to know your own question? Okay, go ahead.
I can't read them, so...
Oh, no. Okay.
I think the middle one is a very interesting one, because you showed with MRI a volumetric change, of course, in the hippocampus.
Yeah.
And normally these changes occur over a long period of time. A year follow-up is normally needed to see a robust change. So how do you explain that can occur so fast? Mm-hmm. What's your opinion about that?
How can I explain it?
What's your view on it? Yeah.
Yeah. Other than saying that it really promotes a lot of different things going on. As I show in my talk, in the animal models, this not just this approach, not just causes reduction of pathology, but really promotes synaptic density. Mm-hmm. And recently, we have some unpublished data also indicates that the stimulation can promote, the health of connectivity, axonal processes, and some other very interesting fundamental brain physiology. So yeah, I mean, this preservation of brain volume in humans is not just observed by us, but other entities doing human trials also observed preservation of brain volume. So I think this is something very fundamental to this idea of increasing oscillation power in the brain.
Thank you so much. Ronald, maybe you have a question, and then to finalize it, one last question in Slido. We're running out of time. Yeah, go. No. Yeah, just start talking, and then they will...
a great talk, Li-Huei, very interesting. And I was triggered by the fact that you show beneficial effects of gamma in many systems, and you say there's many cell types also involved. There's effects on neurons, on microglia, astrocytes. I didn't see the data. It would be very interesting to see the effects on astrocytes. Vasculatures, probably endothelial cells. So how does gamma mechanistically link to these cell types? And more importantly, how to all of them simultaneously?
It's a great question, and we are right now investigating. I think that maybe these different cell types are recruited possibly through different mechanisms. We think that when neurons are recruited to the gamma oscillation range, they could release chemicals and neuropeptides more efficiently. And therefore, different cell types are recruited. We have some evidence for microglia, how they can respond to neurons involved in gamma oscillations differently than neurons not involved in gamma oscillations. And Mitch Murdoch, in fact, currently is working on mechanism how vasomotion is promoted by gamma oscillations, and he has some preliminary observations on the potential molecules that can trigger this response. So, so hopefully in the near future, we can provide more mechanistic insight on this.
Yeah, very nice.
Thank you so much. So we're moving on to the final question for now, and we have more coming in, so we'll try and find a way to send them to you or make you discuss them a little more. Just who's the last one?
I pick out the one of Willem de Haan. He asked the question, to maintain neuronal circuit function, one could argue that an increase of gamma is not necessarily better. Neuronal firing needs to be adaptive to different conditions, and how does this fit with the adagium more gamma is better? I think it's an interesting one, yeah.
Yeah, that's definitely interesting. I don't necessarily know how to answer. I think gamma covers a big range of frequencies. People can say, oh, gamma covers anywhere between 30 to 120 hertz. And the frequencies we are using right now covers so-called low gamma oscillations. So, 40 hertz, between 30 to 60 hertz is the range we are looking at. So I cannot speak for any frequency band beyond the range that we are looking at. And so far, with this low gamma sensory stimulation, we have not seen any detrimental effects.
Thanks a lot. So thank you so much for joining us, Ms. Tsai. Where are you now, by the way?
I'm home.
Yep,
I just got up.
You just got up, yes? Well, what time is it? What time is it with you?
Probably 6:30 or something.
Thank you so much. And you're so bright already. But in what city are you?
In Cambridge, Massachusetts.
All right. Yeah, Cambridge, Massachusetts. That's a different one.
That's a different
different time difference also. Thank you so much. You really sparked an interest there. I can see that, hear that we have still many more questions to ask you. That's a very good sign. Thank you so much.
Thank you.
Thank
Bye.
you.
Thank you.
Yeah, you can wait.